new trends in research and utilization of phytochemicals. So I will talk about around 40 minutes. My best, I will try to give the time. About it will not be like you may not get a very new knowledge. Maybe something it will be you have already heard in seminar and maybe the conferences and from the classes. But I will try to make it from my perspective for the last almost 15 years. What I'm doing the research on phytochemicals and what I think how it will how it is going now and how it should be changed in the future. So this will be my aim of this seminar. So Dr. Nitin has already introduced me, so I'll just skip this. So today I will basically talk about medicinal plants, phytochemicals, and human health, role of phytochemicals in drug discovery. Because this drug discovery research is a very important thing. Because whenever even we talk about the functional foods or nutraceuticals or other things, even in cosmetics, we need a similar type of research. Not that much like a drug discovery detailed research, but we still need from basic to the advanced research, even in the clinical side. So that will probably give the basis for the functional food development. And I'll just talk a little bit about the what are the challenges of phytochemical research, and I will introduce about our research activities, few results I will present, and finally I will go to phytochemical based functional foods, nutraceuticals, and cosmetics. And I think is Dr. Nitin and you all belong in the pharmacy institution, so I thought maybe it is a good chance for me to introduce about pharmacy education in Japan. So to begin with, so medicinal plants are integral part of human health, and we all know that especially in South Asia there is Ayurveda, in China there is a traditional Chinese medicine, in Japan here we use traditional medicine something called as Campo. So medicinal plants are an integral part of this traditional medicine, but not only that, they're also the important source of modern medicines or modern drugs, and we daily use them as a food, herbs, spice, and food additives. They are in essential oil, aromatherapy, fragrances, and especially the new functional foods and cosmetics. And even in the recent year there is a lot of development on plant derived, or what they usually call the green nanotechnology. But one thing we have to consider when Dr. Nitin was introducing, he was talking about underutilized plant resources, was that this was a data from 2017. There is a report you can download and read the report, state of oats plant in 2017, and last year they even published state of oats plants and fungi. So if you see the report, it says that around this more than little bit more than 28,000, but we have 28,000 medicinal plants recorded in the different parts of the world. But total plants there are 4 lakhs, around 4 lakh 60,000. Sorry, for like 80,000 plant species are there. So this means that probably only 6% are used as a medicinal plant, and there is still more than 90% which are not used as medicine. So that doesn't mean that they can be used as medicine, because many of them might be toxic or may not have any activity. But they may have not as a medicine, but they may have potential to be used in other things. So this is one area we should really focus, not only on the medicinal plants but also on the other plant sources other than medicinal plants and their phytochemicals. And today I will not talk much about traditional medicines, because I think this is covered in many lectures and seminars, I think. But one thing we have to keep in mind in that is still around the data says that around or more than 80% of the world population is still depends upon traditional medicine as a primary healthcare. So in that area also phytochemicals are very important. But in traditional medicine especially, not many traditional medicine in not many traditional medicines, their compounds are not identified, and their activity or like pharmacological clinical efficacies are not well studied. So next, this is just a brief introduction plant derived modern drugs. So there are many examples from morphine to quinine, ephedrine, cocaine, reserpine. There are many. There are hundreds of drugs which are derived from plants. So I'll not go much in detail, but there is one interesting report published last year. This is about the last almost 40 years. How much is the role of natural products in modern drug discovery? So this is a paper from Journal of Natural Product published last year. So if you are interested, you can see this paper. And this area, which is in the green, this green and pale green, this is from the natural products. Okay, but this is not only the plants. This is all natural product sources, from microorganisms to marine organisms to animals and other sources. But if we combine this, there is almost around 33% of the modern drugs which were approved in the last 40 years. They were derived from the natural resources. But one thing what is happening in the last 50 years is that first I would like to go to this the graph in the right side. This is the when we talk about natural product, what are the sources of the new drugs, either plants or fungi or other things. Okay, if we see in the totality, this is from Drug Discovery Today, published in 2015. So that might be a little bit old, but it is interesting that if we see in the totality, around 47% of drugs are derived from the plant origin. That is here in the green. But if you see year-wise, in around just like before 1950s, that time most of the drugs were obtained from the plant resources. But if we see in the last 60 or 70 years, the even the drug approval is also less, but still the number, the percentage that plant phytochemicals occupy for the as a source of new drugs is very very less. So about that challenges I will talk later. But one interesting thing about natural product is that this is also another paper from here. You can refer to that if you are interested later in detail. So this is the chemical library what we have till now, and this is the natural product library what we have. But if we think about drugability, or can we make them as a drug, they say that if you see the whole library of synthetic compounds and whole library of natural product compounds, the possibility of natural product to be a drug is high, maybe because they are of the biological origin, or they are biosynthesized, or maybe they are more close to biological system or human physiological system. So still there is a high potential to use natural product in the drug discovery. Just a minute. And there are many screening strategies, like random screening is there, ethnopharmacological approaches are there. I think in India and Nepal, even in Japan, we are using more on this one because we see the traditional medicine and we work on their chemical constituents or biological activity. And there is also biochemical systematics and chemotaxonomy. For example, if we find one potent compound from one genus or one family, we go to search for the similar compound in the members of same genus or same family. And recently there are in silico and computational approaches, and now we have high reverse pharmacological approach and high throughput screening also. But even with the technologies development, there are many challenges, and one challenge now is that like maybe last before 20 or 30 year, it was very easier. People were doing research on any parts of the world. You just go and collect the plant and bring to the another country, another institute. You work on them, you make a drug, you patent. It was like sometimes they also say like what they call the biopiracy and everything. So this was the case. But around in 1992, when the concept of this convention of biological diversity started, and in 2014 when this Nagoya protocol was accepted, or it became a legal aspect, now we are hearing a lot about access and benefit sharing and conservation of biodiversity and the genetic resources. So, and especially for the developing world and for the countries which are very rich in natural resources, this is a good thing, and that at least they can have a share of their own genetic resources. But for a developed country, for example, it is very difficult to bring anything from Nepal, let's say my country, or anything from India and work in Japan. So the challenge here, what I mean is that maybe in Nepal or in India, maybe we do not have that many funds, or we may not have that much facility to the research that the developed countries have. Some institutes are very rich, I know, and very equipped, but in many institutions we don't have that much facility that developed countries laboratories have. So sometimes it makes impossible to do the research in another country's resources. Also, we have problem in plant collection, authentication, cultivation, and main problem is that lack of diversity among new compounds, because most of the plant derived compounds are just their methylated derivative or glucosyl derivative or something. To get a new structure, new carbon backbone is very very difficult. And of course we have resupply problems, and also incompatibility of crude drugs with the high throughput screen. This one I will talk a little bit later. So now I'll go a little bit to what research we do. Basically, my research is mostly focused on the isolation of phytochemicals. This is what I do. We have, let's say, plant or crude drug, then we make an extract, we do column chromatography, and we do an MRMS, and we get the compound, we identify the compound. and we do the biological activity. And another one is like most of the time we also do the biassay guided isolation, where we first check the activity of the extract, and if the one of the extract is active, we go for the fractionation, and if any fraction is active, we again check the biological activity, and we try to get a potent compound. But this is something, it became like a more old method, and nowadays there are many like LC-mass metabolomics here, there NMR metabolomics here, there are many new methods, new techniques which are coming. And even this one is from one of our book chapter we published, like there are many factors who also affect the phytochemicals concentration, and there are many optimization strategies from collection to drying to extraction to purification. And nowadays we'll hear a lot about the green extraction or like advanced extraction techniques. So this is where this is now changing, not only the maceration or like percolation or succination. There are many like microwave studies there, ultrasonic extraction is there, which are more efficient, and maybe in many cases maybe more cheap when you work in the large scale. These things are there. And with the developed technology now, even we can see that there are many uh, studies are there. We can just imagine, for example, this is of turmeric, curcuma lunga. This is a transfer section. So it was in collaboration with one university in Germany, that we can just visualize the compounds there, including curcumin and everything. We can see where the compound is localized, how much is probably the concentration there. So there are many advanced technologies. So we don't need this type of studies now for everything. Okay, but when you talk about the new compound, again the getting them in high amount and getting them and isolating them and doing the NMR, a pure compound, or maybe X-ray crystallography is still important aspect. So till now I have worked this, I'm writing here 2007 because I came as a master student here in Kumamoto University. So I have worked in many medicinal plants of Japan. This is just an example. It's a flower of citrus plant. And we have worked on many medicinal plants of Nepal. But not only that, from Africa, we are working on medicinal plants of Sudan and few medicinal plant of Nigeria also I was working before. And it was plants from the Myanmar also. This was quite interesting. It was in collaboration with another institutes in Japan and Myanmar, that this is one parasitic plant from the Rafflesia family that has the biggest flower in the world. So this genus is called Sapria, and our work here was the first work on this plant to identify the anthocyanins, which give the red color, and also the other phenolic compounds. So my work is mostly focused on polyphenolic compounds, and this was from Bangladesh. So this is called as Banada, or this is Gingiber montanum, like a type of wild ginger in Bangladesh. So I had one PhD student, so he bring this plant to me, and he told me that this plant is delivery. So we derivatize them using cyclodextrins, and we optimize them to make it better soluble and better to have a better pharmacological activity. So we are also doing some work on this type of pharmaceutics related or drug delivery related work. And this one, another one is like we are working now on high altitude medicinal plants of Bhutan also. So this is basic what I am doing. So I'll just give two examples. One is this is a citrus plant. This was in collaboration with one of the city, or let's say municipality in Japan. So this is a special product from there, but there was no scientific study. So they use this green. It's more like a lime, so sour type of orange. So they use with the fish recipe, or even they drink with they just make a slice and keep in some alcoholic drink, and they drink, and they say this is good for health. But there is no any evidence or no any study. So we collaborate with them, we made the extraction, and we check the anti-inflammatory activity. But it was a cell-based study, in vitro study. And from that we also isolated the compound, and we found that some polymethoxyflavonoids had good anti-inflammatory activity using the derived neuronal cells. So we are trying to do this study again now using the mice, the animal model. So this is one study. So if you are interested, you can, this is published in Journal of Food Biochemistry. And another one study we did a few years back was about the matcha. This is also tea. So this is how we usually grow tree, even from Nepal and India. Teas are very popular all over the world. So this is usually how they grow the tea in the open sunlight. And this is green tea. This is like what we get, the black tree, the granules black tree. But in Japan, there is another type. They do they for this, what they call the matcha is that they use almost 90% shade. So they try to shade the light. They make it like a little bit. They want to protect from the sunlight, and they grow the tea, and then finally they make the powder directly from the tea. And this one is called as matcha, and it is not extracted, it is directly kept in water, and it is drink what is called the matcha tea, and it is very popular in Japanese ceremonies, traditional ceremonies. But in recent year it really got like more like a boom, and you can see so many products in Japan. From even KitKat, I kept a photo of KitKat because I think everyone of knew it is bitter, but it's still very popular. So we wanted to know about the scientific evidence and what are the compounds different there. And then they say that this tea, especially matcha, is good for the managing anxiety, or has anti-stress activity. So we use mice model. This is I'm sorry, I forgot to write. This is elevated plus, what we call elevated plus maze test. So this is one. We keep mice here and we just record their movement by camera. So when we administer the matcha 50 milligram and 100 milligram per kilo, we found that these mice, they move more to the open arm. So the time they spend in the open arm and the distance they travel was high. So we somehow found that the powder itself has anti-anxiolytic activity, anxiolytic or anti-anxiety activity. Then we wanted to find that does the water extract, like what we call the green tea, has more activity, or powder itself may have the more activity. Then we selected two methods. Like one, we did like water extraction at 80 degrees centigrade, hot water extraction. So this is more like a green tea. And also we extracted to get a relatively non-polar compounds. We did 80% ethanol extract, and we checked the activity, and we found that both water extract and the 80% or hot water extract and 80% ethanol extract had the activity. But very interesting thing was that when we combine the water extract and when you combine the 80% ethanol extract, they had a higher activity. So one thing we guessed, we hypothesized, was that maybe there is not only a single compound, and which is very common with many extracts, that we may have a different compounds and each of them are like supporting in activity, or what we call the synergistic activity. So this is what we found. And then we opted for the further fractionation of 80% ethanol extract, and we made the hexane soluble fraction, ethyl acetate soluble fraction, and water soluble fraction. And we found that ethyl acetate soluble fraction was had the highest activity. But still, when this ethyl acetate soluble fraction, hexane fraction, and water fraction were combined, they had the better activity. So again, here we found that not only one fraction, but when we combine the fraction, they have a better activity. And to find the mechanism, we used a dopamine D1 receptor blocker, and also the serotonin antagonist. And we found that when you use dopamine receptor blocker or serotonin antagonistic, the activity of matcha was lost. The anxiolytic activity was lost. So we 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 propose that maybe the activity is through the activation of the serotonin receptor and like the dopamine receptor. So when you use them, maybe we lost the activity. And this study is published in Journal of Functional Foods. And you can like the when we in Mr. Matcha, maybe it activates dopaminergic system and serotonergic system, which results in the anti-anxiety level. Okay, so these are two examples what we are working here. But why I'm especially explaining these two activities is that, for example, based on this study, I can easily say that maybe matcha may have a very good activity, and maybe it's good for anxiety treatment or anything. But we write even in title in mice, because if we only write anxiolytic activities of matcha, it will be something like a misleading, or it is very difficult to take this study or extrapolate in the human. So when we talk about drug discovery, I think there are many pharmacy students or professionals, so you know that there is a research and development, preclinical studies, clinical trials, and the review and approval. So, or I will show you one more slide. So this is the drug development process. This is one from the Japanese organization. So we have basic research, non-clinical studies, and then clinical studies and new drug development. So what usually happens with phytochemicals, and I wanted to talk today, is that just based on the in vitro study or just based on the in vivo study, or even from the last year's this COVID infection, only even the in silico studies, okay, or in silico studies are just based on the docking. Many people are proposing that maybe this is the treatment for this disease or that. But one thing we have to consider is that the two results what I have presented you just now are, one is around here, one is a very basic study, okay, it's never goes on the drug development process like that. And another one that we did in mice was also the non-clinical study, somewhere here the basic research. So the results from this type of basic research, and sometimes people say like only on the based of this data, of course there may be a potential, but these products, even for the functional foods, even for the cosmetics, nutraceuticals, they are marketed as like really having a efficacy. Okay, and we have to complete these steps to really prove their efficacy. Okay, so only especially for young students or young professionals, what I want to highlight is that in vitro activity is never equal to in vivo activity, and we should not think like it will have the same activity. And again, in any activity in any animal, maybe in the rabbit or mice or rat, that is not the same activity in human. Okay, so these are completely different. So maybe there might be activity, but we should not extrapolate to make them is like it's highly activity in human. And another thing what we usually say is that, for example, this plant extract has some type of activity, and they say like it's useful in the treatment of diabetes or something like that. Let's say one there is one plant extract or a compound. I also do that type of study, which is active is a alpha amylase or alpha glucosidase inhibitor, only a enzyme inhibition in the in vitro is activity, and we just write it like it's anti-diabetic drug. Okay, it's completely different thing, and we really have to be very careful when we talk about activity and when we talk about the efficacy. And another thing I wanted to highlight is that uh, the use, what they call it, like for example traditional use is a different thing. Okay, for example, I may have a diabetes and I may use something, or I may have a hypertension and I may use something, but that doesn't mean that that is treating me. Okay, using and treatment is two different things, and treatment and cure are two different things. So many in many ethnopharmacological studies, what I usually see even from our country is that like local people use this plant for curing diabetes. Okay, this is not true. They use in diabetes, maybe in the treatment of diabetes, but that doesn't mean that that was really active in the treatment of diabetes. We need research for that. We need to measure the blood glucose level before administration, after administration, and other things. Okay, and curing is a totally different thing. So we should not make it like we should be very careful when using the terminologies. Okay, and another thing what is started more like it became from last year boom is that anything that is natural or herbal, it's safe, and people say like this is a modern drug, this has many side effects. For modern drugs, we know they have side effect because they are studied. They are in the whole this drug development process, they are studied about the possible pharmacological activities, possible side effects. So we know that they have, probably they have some side effects of drug interactions. But for many traditional medicines and natural or herbal products or even phytochemicals, we never studied about their toxicity, we never studied about their safety and other things. So it's not like they may be safe, they may not be safe, but we don't have the evidence. Okay, so as a pharmaceutical professional, we should be very careful when using the terminologies. And another thing really very one serious thing about these problems is, especially with the in vitro studies, is that there is it okay now? Can anyone answer? Because it looks like the everyone was muted.
Yes, sir, it's
Okay.
audible now.
So up to where was audible? Previous slide was audible?
Yes, sir.
Okay. Okay.
Yes,
Sorry,
your
maybe.
slide were audible. Yeah, you can
Okay.
present now your slides.
Yes. So now slides are not visible now. Just a minute then. Just a minute. I will. Sorry for the interruption. So maybe there was some problem with the connection or anything. Is it okay now?
Yes, it is
Okay.
visible, sir.
Is it visible? Okay, thank you. So I was just talking about bioavailability and metabolism. So many phytochemicals, they have to go through the similar process of metabolism is related to the other drugs what we usually study for them, and in many cases they do not have that much scientific evidence for their higher viability or their biological activity. Okay, so I'm just giving example which are very common compounds. There is a curcumin from turmeric or haldi, and there is a resveratrol from red wine, and there is epigallocatechin gallate from the tea what I was talking about. So you can see there are thousands of articles. Today morning only I saw in the PubMed for curcumin there are 17,000 documents. For resveratrol there are thousands documents from epigallocatechin gallate. But if you really see the clinical studies, or even you see the animal based studies, their viability is very low, and they are highly metabolized in the liver and other systems in our body. So these things are also we have to consider. And it's not only for the drug. Okay, I'm just keeping drugs or as a modern drug, because even for the nutraceuticals or functional foods or cosmetics, they have to show the biological activity. That means that maybe only for cosmetics maybe we administer them through the local route or maybe skin, but if you really want them to have some activity in the skin cells, they have to penetrate through the skin and have the activity there. Okay, this is different type of viability. But every molecule we expect every phytochemical to have certain biological activity in our body. They should be absorbed in our body. So this is one aspect many times as a researcher we neglect. So this also we have to take in consideration. And another thing about phytochemicals is that maybe I'll just I use a pointer. So maybe you may have already know about that. There is something called pains in natural product based drug discovery. So this is pan assay interference compounds. So compounds like a curcumin or like quinones and other compounds, whatever in vitro assay you use, they will show very high activity. That is what I was talking in challenges, like they are not compatible with the STS high throughput screening system, but in vivo and clinical studies they may not show the similar activity. Okay, so these are the challenges, and these challenges are same for functional foods. They are same challenges for the nutraceuticals. And another thing is the adulteration. I will not go much in detail, but this was one study published 2 years back, almost 2 years back. They did some survey like how much is the adulteration, and you can see that in the Asia, this green is in the authentic products, the red one is the spurious product or adulterated product, and many all over the world. In Asia, there is more authentic products, and in Africa, what we see from the data, but still all over the world there are many adulterated products. So this one also, when we talk about some formulations, we have to consider. And again, if you are interested in doing phytopharmacological research, I really recommend you to read one article that was published in Journal of Ethnopharmacology last year. And this was by the chief editors of many big journals in phytopharmacological research, ethnopharmacology, phytotherapy research, phytomedicines, and other things. And they have given something like a guideline. So if you read this paper before starting your research, I think it will be very useful. So by summarizing what I have talked now, this phytochemical research is very interesting, and in our countries we have a lot of resources, so it's also interesting. But you really need a very big knowledge of especially the taxonomy, because many articles, many research we see there is a problem of misidentification of plants, and many people do not make the herbariums or boundary specimens. And we really need a very strong background of chemistry to be a phytochemist, and also we need a very strong background of pharmacology and toxicology. I'm sorry for this typo. So this is one I who aspires to be a phytochemical researcher. I really suggest you to have a very good background on these subjects. Okay. So now I'll go a little bit about the second part, the nutritional supplement market. Everyone knows that even from last year's COVID condition, the supplement market has skyrocketed. They are thought to provide adequate amount of essential nutrients for proper functioning of human body. And there are many vitamins, proteins, herbs, meat supplements, sport nutrition, and other products. And in many cases, polyphenol are the main compounds here there also. And there is an increased revenue. There is $1 billion market in that one. Okay. And the terminologies like nutraceuticals, functional foods, some are there. There are also controversy that many people say that we should not use the term nutraceutical or functional foods. There are like some skepticism about them, but there has been a lot of research on that, and many products have also been proven to show the activity. So especially this term nutraceutical was coined in late 1980s, which combines nutrition and pharmaceutical. But this terminology, something called is like a functional foods, was started in Japan, that Dr. said in the beginning in the introduction around in 1980s. And these are the food products. Basically, they are the foods, but they have some beneficial biological activities, or like a more like a pharmacological activities. And this research on nutraceutical has really been very, very high in the recent years. For example, I was writing one book chapter and I did just a survey in Scopus, like nutraceutical or nutraceuticals, and then I just searched and you can find that in the last year only 2020, it was in December, so may not have the all reports from that year, but we had almost 205,000 documents, and mostly these research are from United States, Italy, China, India, China, Canada, and other countries. Okay, and it is, I think in 2020 it will be even more. So this is how they are doing the research. And especially if I talk about the Japan, because we said this concept was started in Japan. Actually, Japanese government's agency, they started this food with the health claims. It was started in 1991. It's officialized in 1991. So there are foods in general which do not have, we cannot write any labeling for them. And there are the foods with health claims which may have, you can write like this may have some activity. And of course there are pharmaceutical product and quasi pharmaceutical product. But within this food with health claim, these two were started in 1990s. First one is foods for specialized health uses, and second one is foods with nutrient functional claims. They were there till they are still in the market, but there were only these two categories till 2015. But in 2015, government again started another category, is the foods with the functional claims. Actually, what happened is that these two products are very difficult in the development, and only big companies can do that, because there needs a lot of funding and a lot of manpower and a lot of research to develop them. But the government's aim was that maybe this another category, foods with functional claims, can be promoted, because to make this labeling you do not have to do research by yourself. For example, there can be a clinical study in one of the part of the world, so you can use that data and you can label your product. But one thing essential in that case is that, for example, if you are making any extract, for that you have to claim that which compound in that extract is active and how much of that. For example, if there is a product which contains quercetin, let's say for example a routine, you have to write that this product, each 100 gram contains 50 milligram of quercetin, which is the active compound for, let's say, lowering the lowering the blood pressure, or maybe lowering the high blood glucose level. Okay. But one thing is that this labeling in Japan is very, very strict, and even functional foods that are targeted for diabetes are not written like this is for diabetes, or this treats the diabetes, okay, or this treats hypertension. Basically, they are more written like this reduces the absorption of glucose, or this reduces the blood glucose level, or this reduces the high blood pressure, or maybe patients with hypertension may get help from this product. So there is nothing like this is the treatment or this is the cure for the diabetes. So this is very important. And this one also is from the same agency, the consumer agency. They said that these are the basic concepts in development of functional foods. First thing is that we have to ensure the safety. Okay, this is the most important thing. And second thing is that we have to have scientific evidence to make the functional claims. Even we say like it will lower your blood glucose level, we really need evidence for that. And the labeling should not be uh, uh, or misleading. Okay, so we should provide the consumers with the information through the proper labeling. This is one thing. So this, and government really reviews the documents, and there are a lot of products with this food with functional claim now, but still government reviews them. And one basic difference is that these upper two products are controlled by the Ministry of Health basically, but this is controlled by the consumer agency. So this is a little bit different aspect in that one. So again, I will come to the same slide. So not only in the drug development, but even in the functional food products and nutraceutical, we really should not forget about that. We should not extrapolate the activities from in vitro or in vivo to the activity in human. Okay. So up to here. Then finally, in the two slides, I will talk about the cosmetics. And we have a long history in South Asia of using plant-based cosmetics from henna to there are so many things, and they're really beneficial in that. And this article, if you, any one of you are interested, it was published last year and I found it quite interesting. It is not my article. So this I really found very interesting. So there are so many compounds in the plants, both primary metabolites and secondary metabolites, that can be used as cosmetic products. And this article also gives some of the example, like different plant parts are used in cosmetics, from bark to flowers to fruits to leaves and nuts and many things, and what may be the potential compound, what may be the cosmetic application. So, and also Japan, Korea, these countries are really big market for cosmetics, and there has been a lot of research, and they are also looking for new resources for the development of these cosmetic products. So this was about what I was talking about, main topic. Now in the last few minutes I will talk about general pharmacy education in Japan, because I think there may be few pharmacy students. So the pharmacy education in Japan has a very long history. So if you want to know more about that, maybe it's a downloadable document in Google. So if you just search annual report of Japan Pharmaceutical Association, you can find this document very easily, and really interesting report. So I'll just share a few pages of that. So in Japan, the overall undergrad till undergraduate, the education is almost same. There is 12 years of schooling. So there is a elementary school, there is lower secondary school, and upper secondary school, or what we call the high school. So up to ninth class, it is a compulsory education. They have to go, but after high school is becomes more like optional. And in pharmacy education, especially, we have now two different type of course. One is 6-year course, what is more close to PharmD in US or other countries, and they have to do the training also. And if they go for 6-year course, they can appear in the national license examination and become a licensed pharmacist. Okay, but another 4-year course is that they do not have the internships in hospital or community pharmacy, and they cannot appear for the license examination. And these two categories are defined because there are students who really want to be a clinical pharmacist or pharmacist as a professional pharmacist, and there are also students who want to be a researcher. So if a student wants to be a researcher, he can go to a 4-year course, then masters, and 2 years master, 3-year PhD, and they can join industry or other organization as a researcher. But if there is a student who is in 6-year course, they can appear in the national license exam, or even after appearing national license exam, they want to go to the graduate school. There is a 4-year PhD. So there is no masters. There is 6-year bachelor degree and 4-year PhD degree, or 4-year bachelor degree, 2-year master degree, and for 3-year PhD degree. And one thing interesting in Japan is that every year there are around 77 schools, I think now, and every year the pass out students are around 14,000. The pass rate is below 70%. It's a very, they say, it's exam is only in Japanese, so there is no chance almost for a foreigner who studied abroad to appear in the exam. But still in the Japanese, the pass rate is below 70%, and it's thought to be very difficult examination. Okay, now what pharmacists do in Japan is, it is a little bit different than what we do in South Asia. Most of the pharmacists, they are in community pharmacy. So they work as a pharmacist there, they operate their own pharmacy. And there are also school pharmacists, so it's in the sanitation. So pharmacists are also involved in the like checking the condition of pools and the condition of sanitization in school and other things. And there are pharmacists in universities who do research and education. There are pharmacists in administrative offices. There are hospital pharmacists who work in a hospital as a core team in the treatment. And there are also clinical research coordinators, because there is a lot of research developing new drugs. And there are also pharmacists in the pharmaceutical industry. So if you see the percentage, now there are around 3 lakhs and 11,000 pharmacists still last 3 years back, and almost 58% are in community pharmacy. So they in community pharmacy, and another 19% are in hospital or clinic. So almost 80% are working as a professional pharmacist, and remaining are in other areas. Okay, and this one is like how the number of pharmacists physicians and pharmacists and nurse, dental physicians are increasing, dentists is increasing. Number of pharmacists and number of physicians is increasing every year. There is some saturation about the dentist. So still in Japan, pharmacy profession is one of the highly respected profession, what I found. And it is also, the income is very high. So there is really a big charm, or like people are really interested to enter in the pharmacy school. And finally, in 2 minutes, I will introduce about our university. Our university, Kumamoto University, is located in the Honshu Island. So maybe you have heard about Hokkaido and Honshu and Shikoku and Kyushu. So our Kumamoto is located in Kyushu. I'm sorry, Kyushu Islands. This is Kumamoto. Here is Fukuoka. Fukuoka is a relatively big city. Maybe you have heard the name. So here we have a pharmacy school. So this is the pharmacy school. I'm in this building now. But my lab is somewhere here in the new building. We don't have in this picture. So here I work in the analytical laboratory. We call it the instrumental analysis laboratory. I work there, and I also involved in management of this traditional medicine museum and the medicinal plant garden also. So in this garden we have around 1500 medicinal plants. So not only research, but we also do like common activities for the common people. And another interesting thing what I found in Japan is that there is the government regulation, or there is a law. It is guided by law that any pharmacy school, every pharmacy school should have a medicinal plant garden. Okay, so this is also compulsory for our school. So we have this one. And I was also involved in making this type of labels, and we are now making database of almost more than 600 medicinal plants. Everything is in Japanese, but we are trying to translate that in English now. So every label has a barcode. So if you click in this barcode, you will be in your iPhone or anything of smartphone, you will direct it to the this page. Okay, so even common people can just visit and access to such information. But not only that, we also collaborate with the local government and public, and we organize events for many school children and common people. Also, we are also involved in making some products. This is a tea made from the tulasi, the holy basil, tulasi, what you call. And also I also do many activities for the junior high school students or elementary school students. So I think this type of science communication is very, very important, because it is important for common people to let them uh, them to know about what research we are doing or what type of educational activity we are doing. Then only maybe our profession as a pharmacist can sustain or can survive in the community. We have to tell them what actually what we are doing and what we are supporting is in the society. Okay, and we are working with many academia, industry, government in the phytochemicals. So we work with the governments, like I was talking about one type of orange. Academia, of course, we are there. We work with the company, and also we work with the community. So this is what we call, like, we are have 4 different partners, and we are trying to make this cycle working now. And finally, I actually realized this because I saw in the poster number 3 sustainable development goal was there. So if you are a phytochemical researcher or plant researcher, you can contribute in many things. So if you are working, please try to connect this to something with the sustainable development goal. Mostly maybe this is related to good health and well-being, but also it is related to the quality education also. It's if you can take the plant cultivation and you can make them uh, if you can connect that to economy, maybe it will be connected to number 1, there is no poverty, and of course number 3, 13, climate action, climate change and other things, and the life on the land. So there are many areas. Not so what I request to the young students is that just don't think about the traditional medicine or modern medicine. Just think about, I will make a new drug, or I will do something about the traditional medicine. Phytochemical research is so broad now that we can find many new areas, we can develop many new areas, and it's a really interesting area. There are like food preservatives there, natural preservatives are there. I didn't talk today much about them. There is a aromatherapy, there is a fragrances industry is there, there is a green nanotechnology there. So there are so many things. So if you are interested, anytime you can contact me. You can search just in Facebook or Twitter. I am